And don't forget to silence your cell phone. The speakers for this session are Dr. Christine Kaye, Ben Shaberman, and John Cornell. Dr. Christine Kaye is a vitreoretinal surgeon at Vitreoretinal Associates in Gainesville, Florida. Prior to this, she was an assistant professor and director of the Retinal Fellowship and Retinal Genetics Service at the University of Florida. She graduated from Harvard University and went to medical school at the University of Florida. She completed her ophthalmology residency at the University of South Florida, her vitreoretinal fellowship training at the University of Iowa. She is the director of retinal genetics and clinical trials now at the Vitreoretinal Associates, involved as principal investigator in 6 IRD clinical trials currently. She has a large IRD patient population with over 800 patients in her clinical database and with 500 of these patients genotyped. We also have Ben Shaberman, who is our Senior Director of Communications for Foundation Fighting Blindness, and John Cornell, our Director of Planned Giving for Foundation Fighting Blindness. Before we begin, I'd like to encourage you to be social with us. You can follow the Visions conversation on Twitter and Facebook and share your experience using the conference hashtag #Visions2018 or #MyVisions2018. This session will be live streamed on our national Facebook page and the Eye on a Cure blog. At this moment, I'm going to go ahead and turn it on over to our presenters and our speakers today. Ben, if you'd like to go ahead and kick us off.
Well, good afternoon, everyone. It's always a pleasure and a privilege for me to be at these conferences and meet you guys and see old friends. It always feels somewhat like a family reunion. And it's always humbling to present with professionals like Dr. Christine Kaye, who's working on a number of clinical trials. I don't know if you know, but John Cornell is an attorney. And I'm a poetry major, so you're in great hands. Remember those tests when you were a kid, like which one doesn't belong? Anyway, on with the program. We're going to break this program into 4 sections, sort of like 4 quarters of a football game. I get the first quarter. I'm going to give a brief overview of clinical trials and talk about 2 stem cell trials, the RenewOn study and the Jsight study. Then I'll hand off, football game, to Christine Kaye. She's involved in, I think it's 6 clinical trials. That's quite a large amount of activity. We'll hear from Christine and her work on those trials. And then John Cornell, for the 3rd quarter, will talk about his experience as a participant in the RenewOn study. And then to round things off, we'll open it up to Q&A. So for those of you that have been at the sessions leading up to this final session of the day, you've heard a lot about all the clinical trials that are underway. And in 1 hour, it's really impossible for us to cover everything. But we put together this text-only sheet of 33 clinical trials that are currently underway for inherited retinal diseases and dry AMD, and Shane will help distribute those at the end of the session. I didn't want to give them to you now because I didn't want you to be distracted while we're talking, but this will be available for you to take away. And we'll do our best to answer about all these different trials, what we know. But please know that you can always visit the blog, Eye on the Cure, on the Foundation's website. You can go to clinicaltrials.gov and obviously talk with the many researchers who are here today. So I think one of the most important takeaways from this session is understanding that a clinical trial is research. And often we talk about people getting treated in clinical trials, and that's sort of a misnomer because something like a gene therapy or stem cell treatment that's in a clinical trial is not really a treatment. If you read our blog or other articles, you'll always see we use words like emerging treatment or potential treatment or promising treatment. It's not a real treatment until it is FDA approved, or if it's being evaluated in Europe, approved by the European Medicines Agency. And so when I say that, what you have to keep in mind is if you want to be in a clinical trial, you're really being a pioneer to advance the research. It's okay to hope that you might have some visual benefit from participating in the trial, but you really shouldn't expect it. You're helping advance the research. And even if a potential treatment doesn't make it through to FDA approval, you're still helping advance knowledge about whatever modality is being studied, about people with your condition. So you're still advancing the research. But remember, you're a pioneer, and I think Dan Chung really talked about the courage and the bravery that you have to put yourself out there as a research subject. In the previous session, you heard about My Retina Tracker, our patient registry. And I'm not going to go into a great deal about it because it was covered in last session. But that's a great way to not only advance knowledge about these diseases, but to learn about clinical trials that you may be appropriate for. So we encourage everybody with an inherited retinal disease to sign up for My Retina Tracker. Likewise, now when you're in My Retina Tracker for a limited time, you have access to free genetic testing through our genetic testing program. And I trust you heard about that in the last session as well. And a genetic test is not only important to getting a definitive diagnosis, it can really help you qualify for a clinical trial, especially if that trial is focused on a specific gene, like a gene therapy trial. So I wanted to get those points out of the way. And let me give you a brief overview of the different phases of a clinical trial. And before a trial even starts, you have those years and years of lab research that lead to a potential therapy, and then the expensive and difficult translational research to move something that looks promising into the clinic. And I don't mean to minimize that by just skimming right over it. There's all that lab research that costs millions and millions of dollars, and only the best things make it to the doorstep of a clinical trial. And when a company or a research group is seeking to launch a trial, they seek authorization from the FDA to do that. And they file something called an investigational new drug application. And the FDA has a certain window of time that they have to respond to that application. And interestingly, if the researchers don't hear back, that means they're okay to proceed forward with their study. And usually in our world, the first phase is a phase 1/2 trial. And that is where people that have the actual conditions are enrolled. And it's usually about 12, 15, 18 people. And the focus is really safety. They are looking at efficacy. They are playing with the dose, doing some dose escalation to see what the highest tolerable dose is. But the real focus is safety. And if things go well, even if there is no sign of efficacy at that early stage, they'll either move to a full-fledged phase 2 or a phase 3. In a phase 2, often what happens is they'll enroll more people. Instead of 12 or 15, maybe it'll be 40, 50, 80. It varies from study to study. And often the people in that phase 2 have better vision. And the hope is they'll see more efficacy either in saving or restoring vision because the patients have healthier retinas. And then if there are some signs of efficacy there and things move forward, that's when we move into a phase 3, and that's the pivotal phase. That's the phase that's really going to make or break the therapy. In a phase 3, you're enrolling even more patients. And one of the key characteristics of a phase 3 is they usually study the therapy in multiple independent sites using the same protocol because they want to make sure that different research teams can come up with the same results. They want to replicate the results. And so if phase 3 goes well, that's when the sponsor of the trial, the company or the research group, will file what's called a new drug application, if it's a drug, or a BLA, biologic license application, if it's something like a gene or a stem cell therapy. And this whole process of going from IND to BLA or NDA approval takes often about 10 years. Luxturna took about 10 years from the launch of the clinical trial to FDA approval. In some cases, it may be a little shorter, but it just takes that much time to really verify that a treatment is safe and that efficacy is sustained. So science, good science, takes a long time. So let me move into a couple stem cell trials you may have heard of already in some of the breakout sessions, the Jsight Trial and the RenewOn Trial. And both are targeting retinitis pigmentosa and potentially Usher syndrome and potentially some related conditions. These were the first 2 stem cell studies for RP. So both use a cell type known as a retinal progenitor. And basically what that is a stem cell that is almost fully developed into a photoreceptor. And as I'm sure many of you know, a photoreceptor is the cell that makes vision possible. So in the case of the Jsight trial, these retinal progenitors are being injected into the vitreous, that soft gel in the middle of the eye. And the idea in the Jsight trial is you're trying to preserve or maybe rescue a little bit the existing photoreceptors, and we call that neuroprotection. So the idea is preserve, protect, maybe rescue a little bit the photoreceptors, especially the cones. And an injection into the vitreous is a relatively low-risk procedure. It can be done usually in a doctor's office, in an outpatient setting. Wet AMD treatments are commonly injected into the vitreous. So it's not all that invasive. So the Jsight trial is underway at the University of California, Irvine under the leadership of Henry Claassen. UCI spun off Jsight a couple, maybe 3 years ago now. That trial's been underway since 2015. They completed their phase 1/2, 28 people were treated, and those receiving the highest dose, which was 3 million cells, their treated eye performed better than their untreated eye. There was evidence that the treatment was showing some efficacy. All the patients had one eye treated, and their untreated eye was the control. So a few months ago, they launched a phase 2B to evaluate 85 patients, up from 25, and evaluate people with even better vision, with hopefully seeing more earnest efficacy. And they're now at 3 sites, even at the phase 2B. They're still at UCI, and they're at practices in Los Angeles and in Boston. And also in the phase 2, they're studying the 3 million cells, the highest dose that they used in the prior phase, and they've even upped it to 6 million cells. They're hoping to see even better efficacy with that. And they're also doing a mock injection, a placebo injection. Good science, you always want a control group to make sure your treatment is performing better than placebo. So then, finally, to round things off, the RenewOn trial started just a little bit after the Jsight trial in 2016 at Mass Eye and Ear Infirmary. It's important for you to know that the Foundation Fighting Blindness funded a significant amount of lab research, including some of the translational research that made this study possible. It's under the leadership of Eric Pierce, who is pretty familiar to many people in the foundation. He chaired our scientific advisory board for 10 years. and he's definitely one of the most accomplished retinal investigators in the world. So again, they're looking at retinal progenitors, but in this trial, they're injecting them underneath the retina. It's called a subretinal injection. And that's a little more invasive. It's a little riskier surgery, or it's actually more riskier than the intravitreal injection. And the reason they're putting these cells underneath the retina is they're hoping that they actually integrate with the patient's existing retina to restore vision, unlike the Jsight trial where you're just trying to preserve what you have. In the RenewOn trial, you're actually trying to restore what's been lost. So in that study, the phase 1/2, they treated 12 people. The treatment was relatively safe. They did not report any efficacy, but now they're launching into a phase 2B, I believe it is, to treat patients that, quote unquote, have a more sufficiently intact retina. So again, they're hoping to see more earnest signs of efficacy. The patients in the 1/2 and the 2B right now have RP, but I read that RenewOn is also seeking approval to study their treatment in people with cone rod dystrophy in this phase of their study. So that concludes my portion, the end of the first quarter, and I will hand off the ball to Dr. Kaye.
All right, good afternoon, everyone. It's great to be here in San Diego. I had a flight this morning from Gainesville, Florida, so it's a long flight, but I'm happy to be here. This is my 6th Visions in a row, and my first 5, I had a Career Development Award here through Foundation Fighting Blindness, which is literally why I'm standing here today talking to you, and literally why I'm in this field and doing multiple clinical trials at my site is almost completely because of the Foundation Fighting Blindness helping launch me into my career. I did my retina fellowship under the tutelage of Edwin Stone and several other wonderful faculty at the University of Iowa, and fell in love with Inherited Retinal Disease Clinic in his on his Tuesday clinics, and applied for a Foundation Fighting Blindness Career Development Award out of fellowship and got it. And Bill Hausworth, who some of you probably know his name as well, was a co-mentor as well as Edwin Stone. And that brought me to University of Florida, where I began working on achromatopsia and the development of AAV therapy, gene therapy for achromatopsia. I've now since had twins and then another little 2-year-old girl now, and ended up moving into private practice, still in Gainesville, Florida, and basically moved my clinical trial practice with me and continued to expand it, also largely because of the continued support of Foundation Fighting Blindness, being able to continue my clinical trial practice. So no more mouse research for me in AAV, but I have a background in that. But what I'd like to talk to you about today is some clinical trials that I'm involved with, and the reasons for talking about this today are twofold. Number one, you may have one of the diseases that I mentioned. We're going to talk about 5 specific, maybe 6 clinical trials if we have time today, very quickly because we don't have much time. So you may have one of those diseases and may actually be taking notes and interested, because I'm going to go through some of the things like inclusion criteria, exclusion criteria, the sites that are doing these trials, and some of the data updates that I'm allowed to give you updates on. Because some things still have not been published or released yet, so I'll only be giving you what data I'm permitted to give you. But secondly, if you don't have one of these diseases, just to give you a flavor and a taste of what some of these clinical trials are like. And just by going through some of the inclusion-exclusion criteria and some of the design of these trials, I'm hoping to shed some light on the clinical trial process. And I'd love to answer questions when we have time at the end for questions on how I consent a patient and the discussion I have with families when I have pediatric patients and parents. You know, this is what I spend my life doing, and what my career is in right now is really inherited retinal disease patients and counseling patients about whether or not you should get into a given, whether I think you should get into a given clinical trial, the risks and the benefits. And informed consent is really a process. It's not a document. It's not even a one-time discussion. It's sometimes multiple appointments. So I'd love to talk to you about some of those things and help guide you. If you have any specific questions about trials that you're interested in, we can talk about that. So first, I'm going to talk about a trial that is going on right now and actually fully enrolled and will be expanding into a larger trial later this year. It's a company called Alkeus. And again, Ben's going to have this distributed at the end of the session, so you'll have some of the names of these trials. But this is a phase 2 tolerability and effects of ALK1, which is the drug, and Stargardt disease, so it's called the TEASE trial. This ALK1 is a modified form of vitamin A, so it's an oral drug that these patients are taking, and there's a placebo-controlled arm as well. And what this drug does, it's a very smart drug. I like to explain this to patients that this drug was designed to address the genetic defect in Stargardt disease, which is the ABCA4 gene. So in patients who have Stargardt, they're lacking an enzyme that processes vitamin A, and they have this toxic byproduct build up in their retina and cause death of cells and damage. And if we could just stop feeding you vitamin A, that might be nice because then that wouldn't build up, except then you'd be blind and you couldn't see because our retinas need vitamin A to work, to dark adapt, to have any rod-based vision. So we can't just deprive you of vitamin A, but what this drug does is it basically reduces the likelihood for vitamin A to dimerize, is the fancy word, and that means bind to itself and form a bad toxic byproduct. It still allows the visual cycle to work, and so patients don't have the dark adaptation issues with some of the other visual cycle modifiers that we've seen in the past with some other trials. But the goal of the drug and what it's been shown to do in mouse models is to decrease this toxic buildup. And you might have heard the word lipofuscin, or your retina specialist, if you have Stargardt disease, might have shown you a colored photograph where you have little yellow spots in the retina if you have Stargardt disease, and that's the buildup of the material that's damaging. And so this drug is the goal of this drug is to decrease the buildup of that. So it's still sort of like vitamin A, but it's super vitamin A. If you think of going to the gas station and get your oil change, it's the super oil product. So it basically decreases the contaminants associated with the processing that needs to happen, which is your visual cycle. And in a mouse model, which was this ABCA4 knockout mouse, their cone response, their ERG cone response, was actually preserved with treatment with this drug. And then in another mouse model that shows faster degeneration, it's called the ABCA4 RDH double knockout mouse. So we have a real fancy mouse model that shows more rapid degeneration similar to a human patient. The changes seen in the RPE were actually slowed or decreased or halted with treatment with this drug. So very good preclinical data in the mouse models with this drug, in my opinion. So as far as the clinical trial, there's a phase 1 multiple dose study that was already completed. Ben talked about phase 1, 2, 3. This trial actually did do a phase 1 first, which means it was done, in this particular circumstance, it was done in patients without Stargardt disease, so just normal patients, anybody. They were just given this drug. Since it's a relatively low-risk drug, you can do that with some trials, and they were just followed for dosing and for pharmacokinetics, and it was found to be safe, and that has been completed and published. The phase 2 was the study that we have just enrolled, patient number 50 at my site actually, and I don't have all 50 patients. I have about 12 or 13 patients at my site. There are about 8 sites in the US, and they enrolled July of 2017 was when the 50th patient enrolled. So if you fast forward 2 years from there, that's when we'll be able to unmask the data, send it to the reading center, and have it have an analysis of whether or not there is a difference between the placebo patients and the treatment patients. Right now we can't actually report, because I've given multiple conferences, this is published data, or not published, but at least publicly available data, that there have been no significant safety events, but we don't have the efficacy values yet because it hasn't been analyzed yet. And importantly, there's been no night blindness reports in the patients, which was something that, of course, is important with any vitamin A drug. And the ALK1 drug is rapidly replacing the normal vitamin A in the patient's blood. There were actually no significant dietary restrictions required, other than patients were told not to fortify excess vitamin A, and they were told not to eat liver. Apparently liver has very high levels of vitamin A, but you could still have carrots, you could still have your normal diet, and then you just had to be taking the pill or the placebo. The primary outcome measure is an efficacy outcome measure, because this is officially a phase 2, and that's autofluorescence, which is a pretty picture of the retina showing how much atrophy or cell death there is. And so we're looking at a 24-month outcome measure of change in size of the atrophy, the growth of loss of cells in the patients who were given placebo versus the patients who were given drug. And the breakdown of it was that 60% of patients the first year of the trial had the drug and 40% had the placebo. And then the second year, half the placebo arm patients flipped into getting treatment. So by the end of the 2 years, 80% of patients had treatment with the drug, and still 20% only had placebo. These are big things that we talk about at the beginning of the game here before we get into this, if a patient is okay with that concept, because you have to be okay with the idea of having a placebo in some trials, particularly oral drug trials will often have a placebo arm. Trial number 2 and 3 are AGTC-sponsored clinical trials. CNG A3 and CNG B3 are 2 different genetic forms of achromatopsia. They're different trials with separate drugs because we have to use the CNG A3 transgene, the CNG B3, so they're completely separate trials and actually even separate sites enrolling in the US. CNG A3, a large population, is in Israel, so we have the Israel site with Eyal Bannon, who's also in that trial. And those are both currently well underway and enrolling. It's an AAV vector that's being used. And there's very good proof of concept data in, of course, a mouse model first, but in this particular disease, there's a large animal model. So there's a dog model for the CNG B3 disease, and then there's a sheep model, which is in Israel, for CNG A3. So basically, after treatment, with one eye being treated in the dog and one eye being treated in the sheep model, these sheep were basically put in a maze, and the way to get a sheep, apparently, to go through a maze is to put all the other sheep on the end of the maze, and they want to get to their friends. And so after treatment, in this disease, achromatopsia is known as day blindness. So patients with achromatopsia are severely light sensitive. They have nystagmus, where their eyes shake. They're completely colorblind, and they have low reduced visual acuity in the 2200 range. But after treatment, the sheep was able to transit the maze at a much higher speed. And same similar scenario for the dogs. They were able to get through a maze at different light levels easier. So the phase 1/2, this is a phase 1/2 trial for these 2 trials. It's a dose escalation trial, like Ben's been mentioning. There's a low, a medium, a high dose cohort. We are in the low dose cohort for both of the trials at this point. The sites are in Florida. You actually have 2 choices: Miami, Bascom Palmer, and then Vitreoretinal Associates, my group up in North Florida in Gainesville, Casey Eye Institute in Oregon, and Chicago Lighthouse. All the patients also travel to Wisconsin for adaptive optics, which is very fancy imaging of the central cones to look at the health of the cone cells that we're hoping to rescue. The CNG A3 sites, also Bascom Palmer, Casey, my site in Gainesville, Mass Eye and Ear, and then Israel. So this is just to give you an idea of some eligibility. So the patients have to have 2, for most, I guess for every gene therapy company that gene therapy trial that I can think of, you have to have 2 proven mutations in the gene in order to be a candidate for that trial. If we're going to be putting you through a surgery and injecting a gene into the retinal cells to try to rescue function, we want to know for 100% sure that you genetically have this disease. So 2 mutations in the gene confirmed, and also importantly, no other confusing mutations in a similar gene. So you can't have another random mutation in the other achromatopsia gene or another macular dystrophy gene that could confuse the diagnosis. So clearly a good genetic diagnosis. We have genetic counselors look over our reports of patients in the screening process to confirm that this is an appropriate patient. And then there's age. So for these 2 trials, the age is 6 and up. We do start in the adult cohort, so we don't get to the 6-year-old cohort until cohort number 4, I believe. So patients, or the pediatric population, is not treated first. You basically come for visits during the first year, and for different companies, you have to also talk about things like lodging and travel reimbursement. These things you always talk to a company about before trials. So, for AGTC, they're paying for the travel for the patients, et cetera, for all of the visits, of course, for all of the surgeries, et cetera. And the endpoints, so of course, number 1 is safety. It is a phase 1/2 trial, so in this case, safety is adverse events is the primary outcome measure. And then our secondary outcome measures are what we believe, after we followed in a long natural history study in both of these diseases, to be points of efficacy that we hope to see rescue in. And so that's visual acuity, color vision, light discomfort. We actually have a fancy, two or three different fancy tests devised to measure how light sensitive patients are and how much pain they're experiencing with light, and then an ERG for retinal function. Number 3 is actually very important to the Foundation Fighting Blindness, is the RUSH-2A trial, which is for USH2A associated retinitis pigmentosa, or which is called non-syndromic RP, or Usher syndrome. So patients with USH2A can actually not have hearing loss. They can just have retinitis pigmentosa, or they can also have hearing loss. JABE Center, which is an IRB center and a research center, is the official sponsor. However, Foundation Fighting Blindness is the collaborator and the original sponsor and creator of this protocol. I don't know if you're aware of this in the room, but there's something called the Clinical Consortium at the Foundation Fighting Blindness, where the FFB in the last year or two has reached out to a number of different clinicians and IRD specialists to create a clinical trial consortium. And we have meetings, and we think of some of the protocols that we should be doing for future clinical trials and devise trials that would make sense for potentially developing new therapies. So this was the first protocol through the clinical consortium. The goal is just natural history observation. So this is the example of a non-treatment trial. We're just observing patients to see how the disease progresses over a period of time. There's 2 different cohorts, and depending on the patient's vision or their visual acuity, they enter one or the second cohort. For this, the patients have to be 8 years or above so we can follow them realistically and do some of the testing we have to do. And they also have to have strictly a genetic confirmation of disease. We don't want to be following, even in a natural history study, a patient who has a confusing genetic diagnosis, who only has 1 mutation, et cetera. And we're following all the outcome measures you would think of: visual field, visual acuity, retinal sensitivity, light sensitivity, et cetera, ERG. And this trial is well underway and ongoing, and we're doing a great job recruiting and the number of sites that are involved in this. So it's exciting to be in the clinical trial consortium at FFB, and we'll continue to expand and be thinking of other trials to do. Number 4, OphthoTech is an injection trial. So moving away from gene therapy, let's think about just injecting a drug in the eye that might help slow the condition. So that's the OphthoTech trial that my site has just started enrolling in. This is for Stargardt disease. This is a phase 2B randomized double-masked controlled trial to look at the safety and efficacy of a drug called Zimura, which is a complement C5 inhibitor. It was actually previously and is currently also being studied in age-related macular degeneration. Not at my site, so I won't talk about that too much, but it's actually nice when I consent the patients to tell them this is not the first time we are putting this in the human eye. There's actually been this exact drug treated in age-related macular degeneration ongoing. The inclusion criteria for the Stargardt trial, age 18 to 50, and that's just what it is. At least 2 pathogenic mutations in the gene. The visual acuity needs to be between 20/20 and 2200. So if you have a patient with 2400, they're not a candidate. And then there's other exclusion criteria. If you've been in a previous gene therapy trial, if you've had a previous stem cell trial treatment, there are exclusion criteria if you've been in a prior trial, if you have a significant health issue, et cetera. Diabetes itself is an exclusion. So it's hard to recruit sometimes when you go through all the inclusion and exclusion criteria, because some patients will have some of these things pop up, abnormal liver enzymes that pop up, et cetera. There's 2 cohorts. Cohort 1 is the Zimura intravitreal injection, and cohort 2 is a sham injection. So again, having this consent discussion with the patient, there's a chance that we're going to put you through this 18-month trial and bring you in once a month and pretend to do an injection in your eye. It's called a sham injection. We basically sterilize the eye. We put some drops in it. We even actually numb it with a needle, but we put a syringe up to the eye and don't actually put the drug into the eye, and that's called a sham injection. And that might sound crude or mean or odd, but that is how almost every macular degeneration trial has gotten their drugs approved, because we can't really put a patient through a trial and allow this placebo effect to really alter the results. So we have to have patients going through the trial not knowing whether they've had the drug or not. And so that's how this company also designed this trial. And the primary outcome measure on this is spectral domain OCT, rate of change in the ellipsoid zone, which is a little line on the OCT over the 18-month period. And then the last trial I'm going to talk about and the reason I'm going to talk about it is because we're going to be a site for that, is the X-linked RP Night Star trial. Interestingly, AGTC is also doing a XLRP trial. So you can look that up on clinicaltrials.gov, but I'm not going to speak about it today because we're going to be a site for the Nightstar trial. It's called Xerius, a dose escalation phase 1-2 trial for X-linked RP, also using AAV, the vector of most common choice. The primary outcome measure is safety, so dose-limiting adverse events and toxicities. The secondary outcome measure is change in best corrected visual acuity from baseline to 12 months. Inclusion, also, we had an interesting discussion about this, I think, in the gene therapy discussion. It's just males. We haven't gotten yet into moving into females, but that's an interesting discussion and potentially something in the future. But males who are over 18 years of age have a genetically confirmed diagnosis of XLRP, and exclusion if they participated in a prior gene therapy trial. They cannot be included. But this is a gene therapy trial that actually is already, and you can look at the website, and this is a publicly traded company, so things have, certain things are of course confidential, but you can see what's listed on the site. But the trial is going well and has already enrolled and treated patients in the United Kingdom. And now they're moving into the United States in the later half of this year and opening up into, I believe, 3 more sites in the United States, and Vitreoretinal Associates in Gainesville will be one of those sites. So in conclusion, and I've probably gone over here, this is really a historic time, and with Luxturna and Spark Therapeutics, amazing job getting that through FDA approval. This is an incredible time for gene therapy, but also for lots of other clinical trials. My only thoughts and advice and parting thoughts would be, if you have not had genetic testing, get genetic testing. There's Foundation Fighting Blindness is funding this now. There's relatively logistically easy ways to get genetic testing now. Hopefully you went to the session before this and they talked to, so if you haven't had genetic testing, make sure that you try to get that. Number 2, see an IRD specialist at least once, at least for that clinic. I know they can be difficult to find. There's not tons of us in the US that do inherited retinal disease, but try to at least at one point see an IRD specialist, if for nothing else but to discuss what clinical trials you might be a candidate for, so you know your options. There's really, and I didn't go into some of these trials, but there's no level of vision that might not be a candidate for something, and I think that that's something that's important to communicate. You might be light perception vision with RP and be a great candidate for instance, the optogenetic study right now, because that's actually the criteria. I'm not involved in that, but I'll just say one word about it. I send a lot of patients over to Dave Birch's group in Texas who have light perception vision with retinitis pigmentosa for their phase 1-2 trial in optogenetics, because that's the inclusion criteria for that treatment right now, and that's the intravitreal optogenetics approach to repairing retinal function. And then lastly, go to these conferences and get involved in My Retina Tracker and continue to support Foundation Fighting Blindness and science, as you already are doing by virtue of the fact that you're here. So thank you very much for your time, and I'm happy to answer any questions when we get to the 4th quarter.
There you go.
Okay, I'm not so good at clock management, so forget the halftime beer and pretzels. We're moving right to the 3rd quarter. Shane introduced me as Director of Plan Giving at the Foundation, which I am, but I'm not talking to you about Plan Giving. Ben mentioned that I'm an attorney. Well, he's not correct on that. I was an attorney. I'm Director of Plan Giving for the Foundation. I don't give legal advice. Even if I was still an attorney, I wouldn't talk to you about anything legal today. What I'm talking to you today about, the hat I'm wearing, is just like you. I'm talking to you personally about what it was like to go through, and is like, because I'm still in the RenewOn Trial, and the similar trials that Dr. Kaye just talked about. What I realized when I was looking at my notes to try to prepare is I used to have everything I'm going to try to tell you committed to memory. And I wasn't nervous. I'm nervous right now. I realize that 3 years has gone by since this process started. So I'll do the best I can to relate to you the process, some of the things that I felt and I experienced when I went through it. I'll tell you right up front, I cannot answer the questions that you want the answer to. These things are very private. We cannot talk about efficacy, the results, because the trial is still ongoing, and it's very critical that when you get involved in one of these, you respect the integrity of the process and wait until the public announcements come. So having said that, I learned about the RenewOn Trial by being connected to the foundation, not as an employee, but by reading the website, the emails that come out. On May 6th of 2015, 3 years ago, was the press release where the FDA announced this clinical trial being launched by RenewOn, which is a British company. I had no thoughts prior to that, really, about being in a clinical trial myself. All I would do would be talk to members, donors, constituents about how important clinical trials were. I did not have as my dream, my lifelong goal, to be in a clinical trial. So that was May 6th. At the end of June the same year, we were at a Visions conference, and guess who was there? Dr. Eric Pierce. And Julie Anderson and I were together, and we were at a dinner, and anyone you want to say hi to? Well, let's go say hi to Dr. Pierce, who I'd never met before. Let's just wish him well in the trial that we heard about. We found him. We waited and waited until everyone else was talking to him and started asking questions and wishing him well. And I said to him, well, what does a person do if they want to get involved in a trial? I was trying to gain information to impart to others like you when asked. He said, well, I think you just asked me. I said, I don't know what you're talking about. So in any event, he thought that I was interested in being a candidate for the trial. So that actually got me thinking about it. So that was the end of June of 2015. July went by, August went by, September, October, November, exchanged a couple emails. I kept checking clinicaltrials.gov. I didn't hear anything from anybody, so I thought maybe they'd already started, maybe they weren't recruiting anymore. And believe me, I'm not being critical. This was just the process. So I finally connected with Dr. Pierce in early December of 2015. We had a telephone conversation where I described my visual functional level in general terms. He said, well, it sounds like you might possibly meet the objective criteria that we have for our trial. If you'd like to come to Boston, where this was being done at Massachusetts Eye and Ear Infirmary, we can give you some tests. So Julie and I went out there for a few days in early January of 2016, did some baseline testing. About the first week in February, I received an email from Dr. Pierce's office saying that I met the objective criteria. In other words, your eyes are bad enough, John. We would like you to participate in the trial. So that's really when the rubber started to meet the road for me. It became real at that point. This was not just a science fiction. There was actually a clinical trial. I'd been to the doctor's office. I'd had the tests, and I've actually been invited to participate. And that's when the anxiety started. So I took a week or so. I consulted with family, some people within the foundation, some names that you know, to really try to do as much due diligence as I possibly could. Dr. Pierce in his office went through all the informed consent things with me when I was there in January. So about a week later, I decided to and did accept their invitation to be involved in the trial. This was like mid-February, February of 2016. The baseline testing was only good for 90 days. So it should have, the surgery would have had to have happened by April of 2016. So I was getting nervous. Why wasn't I getting a call? When am I going to hear from them? And then April came and went, and I knew that 90 days was gone, so it wasn't going to happen. In July, I got an email from Dr. Pierce, early July, saying that the first 2 participants had received their injections. Another dose of reality. It's real. Patients have actually received the stem cell injections. About a week later, I received a call saying that my surgery was going to be set for August 25th of 2016. I sent out emails to everybody, oh my God, the date is set, this is great, this is great. 10 days later, I got a call, the surgery had been canceled. So they still had to do baseline testing. They wanted to do it in early August for the August 25th surgery schedule date. So we went to Boston anyway, did some more baseline testing for a few days, and then they told me that my new surgery date would be September 29th. And of course, I was now thinking, well, it only will happen when it happens. So I did not get my hopes set too high for that, but I didn't get a call that it wasn't happening, made all the hotel reservations, made all the plane reservations, got to Boston on September 26th, the week of the surgery, which was a Monday. Tuesday, more baseline testing. Wednesday, off day. Thursday, the 29th, was the actual surgery date. And by the way, that was the same day of the Hamilton Music event, the FFB fundraiser in Chicago that we had tickets for that we had to give up because of the surgery. On the 29th of September, we're still in 2016, at the hospital by 6:30 in the morning. I was feeling good by the drugs at 7:30. That's the last I remember. I woke up at 10 o'clock, out of surgery, laying on my back for a few hours. By 3:30 that afternoon, Julie and I were walking back to the hospital. That was Thursday. Follow-up appointment Friday morning, follow-up appointment Saturday morning, follow-up appointment Monday morning of the following week, follow-up appointment Wednesday morning of the following week. That's October 5th. Flew home that night. Went back on October 12th, a week later. Went back October 24th, 2 weeks later. Back November 28th after Thanksgiving. And by the way, each one of these appointments there was some commonality. There was always a physical exam. There was always a refraction, looking at the eye chart. Sometimes, or there's almost always an EKG. There was always a blood draw. So they always said, don't take a flight back before 7 p.m. the night of your testing. And that's what we did. And sometimes we'd be out there for 2 days, sometimes 3 days, depending on airfares, depending upon hotel costs, all these things that you had to do really to be respectful of the cost that's involved, I'm going to talk about in a minute. So I'm up to November 28th, still in 2016. Go into January, early January, for the 3-month, I believe, if my math is correct, follow-up appointment. 7 inches of snow landing in Boston, and I'm not a real good flyer. I did not like that day at all. March, went back for the 6-month follow-up. June, went back for the 9-month follow-up. September of 2017 was the 1-year milestone. I really thought that was going to be the end of it. They thought that would be our participation commitment would be 1 year. When I got there, they said that RenewOn really wants to follow the participants for another 18 months to 2 years. Would you be willing to do that? And of course, when you're in it at that point, and one of the biggest things you have to do is that when you get involved in one of these things, even though you have the right absolutely to walk away whenever you want to, you really need to go into it with the idea that you're going to commit to see it through to the end, because you've heard it, we're advancing the research, that's what we're doing. So I had a telephone appointment in March of this year at the 1.5-year mark. Then the last, which I believe will be the last appointment, will be at the 2-year mark. That'll be in September of this year. All of those things that I just told you entailed about 10 or 11 separate trips to Boston, comprising of over 30 days. And there's 2 quick things that I want to tell you, 2 quick stories. So, including with Dr. Eric Pierce on the team, was Dr. Jason Commander. He was one of the surgeons. Could you think of a better name for one of your surgeons than Commander? So, and Dr. Dean Elliott also did the surgery. So well, I don't know what they knew, but I didn't say that I worked for the foundation at the time. I wanted it to be, this is personal. I got this opportunity because I pursued it like anybody else in this room could have pursued it. So about the 3rd or 4th appointment after the surgery, we're sitting in Dr. Commander's office, and I'm sitting at this chair, and he's back to my right at his computer typing away and asking me questions. And all of a sudden he got quiet, and he pushes back and he goes, "You work for the foundation?" I said, "Yes." He goes, "Well, let me tell you something. I wouldn't be in the position where I am today but for the foundation fighting blindness." And he went on to tell the great work of the foundation, how it funded him. And I just want all of you to know that the money that you raise really goes to quality people. Not just quality research, but quality people through our scientific advisory board, the vetting that's done. So, and he said I could say that to you. The other thing about Dr. Commander is he's again sitting at his computer, and one of the first things that they noticed, and they took pictures every single time, is that the 2 days after the surgery, again, they injected these stem cells subretinally, they saw a clumping, like a bump, in my retina. They theorized, but we're not sure, and to this day don't know for sure, that may have been the stem cells that they injected. And they had not seen that on any other of the participants to date. So I didn't know if I was supposed to get excited about that or get bored or what, but anyway, that's what they told me. So another appointment in his office, he said, "John, I got to tell you something. Remember 6 months ago or a year ago I told you about that clumping under your retina?" I said, "Yes, I remember." He goes, "Well, I thought it was so cool I made it my wallpaper on my monitor." And I said, "Well, Jason, that's really cool. I guess, but I got to tell you something. It's pretty nerdy, too. He goes, I'll take that as a compliment. Well, I meant it as a compliment, because you want your science guys to be nerdy, right? I mean, I do. So a big commitment, a big time frame, roller coaster of emotions. The expense involved, it was almost all reimbursed by RenewOn to their credit. So that means airfare, ground transportation, hotel cost, and they provided a stipend of $60 per day for every appointment, not every meal, just every day that I had an appointment that I was out there, $60. Didn't cover everything, but almost everything. And also to their credit, they covered Julie's airfare. I would not have wanted to go through that alone. I'm not sure I would have done it alone. So she, as my companion and caregiver in that situation, they covered her expense as well. And I kept track of it, a little over $20,000, just for what I told you. It has nothing to do with their medical costs or anything else, just the expense for my travel and participation in that. And then the last thing, I'm going to take my time here, since I have the floor, I'm going to give some thanks. I want to give some thanks to people at RenewOn for investing in this project. This is what it's all about. This is what we raise money for, is clinical trials. Because there's no possibility of treatments until it's gone through the clinical trial process. And we need some companies to take the risk to do that. So I want to thank RenewOn for doing that. I also want to thank Dr. Pierce and his team, because this is a scary thing to go through. And I'll tell you that as much as Ben is absolutely correct in saying that these are trials, these are human experiments, we're supposed to think about what we're doing is advancing the research, I'd be lying to you if I said I wasn't hopeful for some vision restoration. I think that's normal. I think it's healthy. It maybe wasn't healthy all the time, but I was thinking about that off and on. But his team made it so comfortable for us that I mean, they were funny, they were professional, they were compassionate, they were punctual, they said something, they did it. So it couldn't have been a better experience for Julie and myself to have gone through, and that's a credit to them, and also credit to the foundation for our association with them. I, of course, couldn't get through this without thanking the foundation. I mean, but for the foundation fighting blindness and Gordon and Luli and his family and the Bermans that founded this organization, we wouldn't be having this discussion right now. We need to keep that in our minds and use it as inspiration as we move forward into the future, these really, really exciting times that we're hearing about. And I want to thank so many people that followed me through the journey, sent me emails, called me. It really did help encourage me. Because after a while, after the excitement wore off, it became a little bit monotonous and a little bit tiring at times. It did help me go through it. And lastly, Julie, you're going to kill me, but I'm going to thank Julie for this, because what often gets lost in these things is the role of the companion and the caregiver. I mean, Julie has RP, and she's suffering through her own vision loss right now. But what she was doing was being there supportive of me through the boredom, through the snowstorms. And if I'm a bad flyer, you ought to see her in action. It is not pretty. But I just want you to know that it's a real human life experience. It's what we're in this journey for. And Julie, I thank you from the bottom of my heart for you being there with me. As I said, I wouldn't have done it alone. So thank you. And thank all of you for listening.
Thank you very much, Dr. Kaye, Ben, and John. Now we're going to take about 10 minutes to take some questions from the audience. Because we are recording this session, please wait until the microphone runner reaches you so that you may ask your question into the microphone. To ensure we get to as many people as possible, please limit yourself to one short question when it's your turn. So please just raise your hands high in the air, and I will try and identify folks. So I already see one back here. Diane, this gentleman in the black shirt.
Thanks. John, appreciate that. I was actually in the late, great Dr. Burson's trial for lutein, so it was great to hear the story. My decision was a lot easier because, as you know, Luton was a simple supplement, if you will, rather than what you went through. But my question really is this: how did it affect you in terms of your decision to do that particular trial, knowing you would likely never be a candidate for another similar type of trial in the future? Or, you know, you committed one path and that was it.
That's a great. Hello?
Yep.
Oh, okay. That question did come up, and I don't think necessarily it's a foregone conclusion that, I mean, there's not a per se disqualification from future clinical trials. I mean, the reality is more than likely it would be a potential exclusionary event. So I did have to think about that. And you know what, really, there were two things that really put me over the edge in this decision. One is I talked to some people really close to the foundation here who knew Eric Pierce from way back when, and they said with their blind son, if Eric was going to be having surgery, we would not want him in anyone's hands other than Dr. Pierce's. So that made me feel really good and confident about. That's not to say that, you know, others aren't equally as competent, but that made me feel good because that's where I was. The other thing was, and this may sound kind of corny, but all I kept thinking about is I keep talking about clinical trials and people have to step forward, and finally it dawned on me, wait a minute, you can do it right now. So let's forget about the hypocrisy. This opportunity's been given to you. It feels like you should do it, so you need to do it. That's about as, you know, as deep as it got, really.
Excellent. And I know we had a question, Patty, over there.
Hi there. My question is on more specifics on finding the patients for the clinical trials. As a practitioner, when you're starting a trial, do you go to My Retina Tracker to find the actual patients? Do you use your private practice to find the patients? Or should we, as the possible patients, be looking for the trials ourselves and then try to figure out how we can get into them?
That is a great question. So to answer your first question, I have not yet, but that is a very good point that I should start thinking about looking at My Retina Tracker for recruitment. That is one of the points of My Retina Tracker. But I have a very large database. I have about 800, 900 patients in my inherited retinal disease database. So I've, over the last 8 years of doing this, collected and saved the information of patients that I see in my own clinic. So most, you know, I usually draw from my own database first. I've been talking about genetic testing for the last 6 years of these conferences. We should all get genetic testing, so I do genetic testing on almost all of my patients. I find some way to get it done. So I have about 500 of them genotyped in that database. So I have a pretty good genetic database ready to recruit from. Then when you get involved in a clinical trial, so first of all, to be a site for a clinical trial, you have to probably have some patients and be able to, when you go through site selection, these companies or nonprofits, whoever's doing the trial, they contact you and they say, do you have patients, do you have X number of patients with this disease within this age, with this visual acuity? So to be a site, you have to have sort of shown that you have some patients. And then you potentially start enrolling. And then the companies or the sponsor of the trial will actually also help you with recruitment. So patients, but yes, patients absolutely should also take the initiative. So the way that some of the patients are coming to me are patient-initiated contact to the sponsor by looking on the clinicaltrials.gov site, where a patient literally looked at the clinicaltrials.gov site and saw contact Jill Dolgen at AGTC, who happens to be here somewhere, and it literally lists her name and the advocacy.agtc or whatever. That just is the first one that came to mind. But usually in the clinicaltrials.gov, there'll be a section on the bottom for contact, and then patients can directly contact either the site or the sponsor and reach out and see if they might be a candidate. And FFB also helps with that. I don't know if Ben wants to sort of talk about My Retina Tracker a bit more, but yes, I mean, to answer your question, patients should definitely also initiate as much as they can and reach out directly and look at the clinicaltrials.gov site. But it can be overwhelming, and you can have, there are multiple trials potentially that you could be a candidate for, so I still think that seeing an IRD specialist and talking with them about whether or not it's a good idea to be in one of those trials is a very important part of it.
All right, we have time for one or two more questions. Any other questions? Raise your hand high.
There's someone over there.
Oh, there we go. Jill, Diane, if you can get a microphone over there. Thank you.
John, I don't think this is on. Is it? Well, anyway, I just want to ask John, do you have RP or Usher?
I have RP.
Thank you. Anyway, thank you all for all your presentations. It's been really, really great.
I wanted to add one more thing to the last question that Christine about finding out. What ended up happening is when I thought really I wasn't going to be contacted, I went to clinicaltrials.gov and found the RenewOn Trial, and at the bottom it said, if you're interested in this trial, please call massioneerinfo@whatever.gov. And I thought, oh, right, it's like somebody's going to respond to that. I sent an email, and they responded to that. And that's really what started the dialogue. So it's worth doing. You have to be your own advocate in any medical thing, but particularly the clinical trial process.
Excellent. And trying to stay on timeline, we do have the one-pager of information that Ben shared with us, and so we're going to be handing those out at the exit doors. We have Connor and Diane who will be standing at the exits. And if you do have other questions, I'm sure our folks up here will be sticking around for just a few minutes. So if you want to build lines, and I'm sure they'll answer a couple of questions. Sorry, I'm putting you all on the spot there. But thank you for attending this session. The welcome reception will begin at 5:30 p.m. in the Grand Ballroom ABC, so where the exhibit hall is at. The next series of sessions will begin at 7 p.m., so please check your program for session choices and locations. Thank you, everybody, for coming today.